This is the euchromatin heterochromatin part. Now, euchromatin is the loosely packaged chromatin that you can access easily. Meanwhile, heterochromatin is the tightly packaged chromatin. Actually, it's so highly compacted that it looks like a black spot in micrographs. The difference between these two parts is you need to make your chromatin compartmentalize in a way that you can access the parts you need the most while you're archiving and protecting the parts that you don't frequently use. This system, the euchromatin heterochromatin system, allows you to do that. Now, let's look at the heterochromatin, the compact part. It has two different types, facultative heterochromatin and constitutive heterochromatin. Facultative heterochromatin can be converted into euchromatin, vice versa. For example, let's say you have a type of pigment coding gene that's used for your eyes. For example, to make your eyes brown. You would not need this pigment in, let's say, your stomach cells or your follicle cells or your epithelial cells. So in some cells, it is heterochromatin. Meanwhile, in some cells, it might be euchromatin. It might be more frequently used. Or perhaps it's only used in embryonic stage and then converted into a more compact archive structure later on. In these cases, this heterochromatin can be converted to and back to euchromatin, which means it's facultative heterochromatin. Constitutive heterochromatin is somewhat what its name is implying. It's constitutive. It's useless. Let's not say it's useless, it has structural use, but it's not something you read and make a protein out of. And it's always in heterochromatin structure. It's never converted into a more loose state. Constitutive heterochromatin also has two types, centromeres and telomeres. But before we come to that, I want to state three things that are same in both centromeres and telomeres. 1, they don't code for any proteins. 2, they're permanently compacted. 3, they contain highly repetitive sequences. For example, telomeres contain TTAGGG sequence over and over again. Now, why do they do this? Because due to the fact we have linear genomes and not circular genomes, we lose a part of our genome every time we replicate it. This is basically a mechanical issue that we cannot fix by fixing the enzyme. So we add nonsense to the end of the genome. So if we lose that nonsense, if we lose that part of the genome several times even, it won't matter. It's just nonsense. It's just TTAGGG repeated for 1,000 times. Centromeres have a little bit of a different function. Centromeres also have highly repetitive sequences, but what the sequence is or the length of the sequence doesn't really matter. What matters is the sequence can interact with a protein, especially kinetochores. Kinetochores are proteins that hold on to mitotic chromosomes from each side and associate the chromosome with the mitotic spindle, which is the string that pulls the chromosomes away and rips them into two sister chromatids. You've definitely seen this in middle school biology class or something. So centromeres have that function. Centromeres also maintain sister chromatid cohesion. So they create that X shape during mitosis, and then they associate it with the protein that pulls apart the kind of branches of the X. Other than that, I want to note this, that in telomeres, the length of the telomere determines how much a cell can replicate its DNA without literally damaging its own DNA, which means our cells are not immortal. When the telomere ends, when it's chopped off so many times that we reach the actual DNA, then we can't chop more, we can't replicate more because we are going to chop into the DNA that we're going to use. So in that case, if we keep replicating, if we keep dividing, we're either going to become cancer, and by we I mean the cell. The cell's either going to become cancer or it's going to realize what it's doing and kill itself, like honorable cells do. However, there are some situations where a cell is immortal because it has an enzyme called telomerase that adds on to the telomere ends over and over and lengthens them as the cell is replicating and losing the telomeres. There are several minus points to this too. but I'm going to get into that later on. Just know that in cancer cells, the immortality, the infinite capability to divide is caused by the telomerase enzyme. Sorry, my tongue's a little searchy. Okay, before we look at repeats in DNA, let me tell you about G-bands. G-bands are the bands we see when we dye a chromosome with a stain like GM stain. GM stain associates with DNA, so where DNA is packaged thicker, it's going to accumulate more and create a darker shape. The dark and light shapes, the bands, the G-bands, as we call them, give the chromosome a unique signature by which we can literally tell if that's chromosome 1 or chromosome 2, or if that's a normal chromosome, or if there's something off with that. For example, sometimes in Down syndrome or some other syndromes, an extra chromosome might be there, or a chromosome might have a leg chopped off, reversed, and stuck back together. And that can cause problems. This is how we detect that. We literally just look at the bands like a barcode. Now I'm going to get into repeats in DNA.